Connaissance.
Recherche.
Sciences.
Des sciences mathématiques.
Théorie.
Physique.
Philosophie.
Conférence.
Archéologie.
Nature.
So I want to thank you, thank the organizers for this very kind invitation. And also obviously the opportunity to talk a little bit of malaria. Talking of malaria in a meeting dedicated to vaccines on the future seems a little odd. I'm sure you understand, since there's still no marketed vaccines against malaria. And the ongoing trials that are ongoing are really not that promising. Doing better than nature seems to be actually a minimum, since, as you know, malaria is a lifelong infection. So people, even though they're not sick throughout their life, they actually carry the parasite for almost all their life. So what is certain is that we really need to learn and do better. So in the lab, we're working on a rodent model of infection, and we're trying to understand how infection proceeds at the very, very early steps of infection, immediately after the parasites are injected by the mosquito. And what I would like to tell you is some imaging studies that we've been doing that are changing our view of these very, very early steps, and maybe how this might provide some clues as to how the protective immunity might be mounted. So just a very quick outlook at the malaria situation. It's still quite bad with these numbers, and in particular 1 to 2 million deaths per year, mostly in children and almost exclusively in Africa. These are the tools that are available today. They're quite simple. They're mostly targeting the mosquito or the mosquito host interaction with insecticide-treated nets and indoor residual spraying, as well as drug treatments, either preventive or curative. And among the tools for the future are vaccines, but I would say that it's rather than looking for a vaccine of the future, I think the need is really to hope for a vaccine in the future. So, why don't we have a vaccine? And a classical reason stated is the complexity of the parasite and of the life cycle. The life cycle can be divided schematically in 3 phases. The first one is the multiplication of the parasite inside red blood cells, responsible for all the symptoms and complications of the disease. Second phase is transmission through the mosquito. And the third phase is the so-called pre-erythrocytic phase. We will concentrate on this one, which basically extends from injection of the parasite by the mosquito to the first multiplication cycles of the parasite inside red blood cells. So each of these phases has or is the target of distinct vaccination strategies, because the parasite has stage-specific products. So you have blood stage vaccines, which aim at reducing the incidence and the severity of the disease, not infection itself. They're based on antibodies that recognize free parasites or infected red blood cells, but so far they really have limited efficiency. The transmission-blocking vaccines are trying to block the transmission from infected men to mosquitoes. They're targeting surface proteins on the sexual stages of the parasite and the zygote, which develop inside the mosquito gut. Again, they're based on antibodies in these protected individuals. No, actually, the individuals who are vaccinated are not protected, and the vaccine is called altruistic because it does not protect the vaccinated individual, but it protects the community by trying to cut down on transmission. And the last type of vaccines are the PEP, or the pre-erythrocytic phase vaccines, which try to reduce infection. They're certainly the most attractive because if they're efficient, they reduce infection and they cut the cycle before red blood cell infection happens, and it is potentially the most efficient. Okay, so what is the pre-erythrocytic phase? So basically it's the life of this stage of the parasite which is injected by the mosquito, the sporozoite. They are injected in the skin, and this has been recognized only quite recently. Until almost the mid-90s, it was thought that the mosquito would inject the parasite directly into the bloodstream. So eventually the parasite reaches the bloodstream, then goes to the liver, invades hepatocytes, multiplies inside an hepatocyte into tens of thousands of a new form of the parasite, which is called the merozoite, and start infection of the red blood cells. Now, the field of PEP vaccines has been dominated by a model of protection, which is induced by live attenuated sporozoites. It was first described using irradiated sporozoite, either with gamma radiations or UV radiations. Basically, the parasite remains alive and motile, so can reach the liver, invade hepatocytes, but then the development of these irradiated parasites is blocked inside hepatocytes, and there is no red blood cell infection. This works in about all models. It's been known for a while, as you can see, 41, the first demonstration in birds, and it works also in humans using mosquito bites, but it takes a lot of mosquito bites, up to thousands of mosquito bites delivered in very heavy protocols over a year time to protect an individual, but eventually can work, and it's a sterile protection. It really protects against the injection of thousands of virulent parasites. You can also protect by using normal sporozoites and treating with a drug like chloroquine that blocks infection in the red blood cells as soon as it starts. And then more recently, genetically attenuated parasites have been described. Basically, they infect the liver, but their development is blocked inside hepatocytes. So what all these techniques have in common is to basically load the organisms with lots of large amounts of parasites, and they do not go beyond the liver, and erythrocytic infection is not induced, and that protects the individual. What is interesting, by the way, is that when protective parasites were just irradiated, it was considered completely impractical for vaccination of humans, and since a few years, since it was used with genetically attenuated parasite, then it becomes very fashionable, and the whole community is very leaning towards the use of this kind of technology for human vaccination, despite the challenges that are still there. We might discuss that. Okay, so the effectors of protection in this sole model of complete and sterile protection against malaria have been investigated in rodents, but after IV injection of parasites. And just about all arms of the immune response have been described, particularly antibodies that prevent the migration of the parasite as well as invasion of hepatocytes, and CD4 and CD8 cells that recognize infected hepatocytes. In particular, CD8 are supposed to have really a key role in protection. It has been shown by a series of adoptive transfer, and they seem to be sufficient to confer protection. The exact way they kill the infected hepatocyte is still not very well known. But it's been shown more than 20 years ago that CD8, recognizing just a single epitope of the major surface protein of the sporozoite, which is called CS, are actually sufficient to protect individuals. So one major question, obviously, was where in this model are the parasites delivered, and where are the CD8 T cells primed? And basically, because all the parasites were thought to only go to the liver, it was absolutely normal to consider that it was in the liver that these cells would be primed, and the idea was that it was a combination of extracellular sporozoite as well as intracellular parasites that whose development leads to apoptosis of infected hepatocytes, and all would go to the lymph node in the liver. But basically, that would be the organ in which the CD8 would be primed and would, of course, become effective. So we're interested in parasite biology and want to understand infection by and the fate of these parasites in the host. And we've been approaching this problem through imaging in a rodent system. It's really logistically quite complex, and you can't do experiments when you want. So you need mosquitoes infected by, we use a species called Plasmodium berghii. It's transgenic for GFP and all sorts of markers. Then we use hairless mice in general, which are quite good for doing imaging in the skin because of the lack of autofluorescence. And we look at the parasite inside the skin and intravitally in different organs using a high-speed spinning disc confocal microscope, which is really the best adapted to follow the very fast-moving objects, and sporozoites certainly are fast-moving objects. So what do we see at the injection site? We see sporozoites. We consider that an infected mosquito usually inject 10 to hundreds of sporozoite. They move in a quite impressive way. It's called a gliding type of motility. It uses an actin-myosin motor, and you can see here the kind of path using maximum intensity projections that show the path of the parasite. You can see these green dots. This is at the 3rd minute post-infection. Okay, so as expected, what we see is that these parasites invade blood vessels. You can see here an example. This is a parasite moving in the dermis that glides and then glides around the blood vessel and then will penetrate the endothelium. It's not clear exactly how it does that, and then it's taken up with the blood flow. The real surprise of these experiments, and we did a lot of these, is that only 30% of the parasites that we can actually image are caught doing this, and only 30% leave the site of bite through the blood. So the fate of these is obviously to go to the liver very rapidly, so they leave the sinusoid, they penetrate in the parenchyma, they eventually infect a hepatocyte inside a parasitophorous vacuole. They transform into these merozoites. One question that has really been important in the field is how are these little parasites released back into the bloodstream, and it was thought that the infected cell would actually mechanically burst. And imaging once again has shown that the parasite is much more inventive than that. So the parasite is in green, the sinusoids in red, and the parenchyma in black. And you can see the actually thousands or hundreds of thousands of little parasites actually moving into what we call a merosome, which is a formation of lots of these little parasites surrounded by the plasma membrane of the hepatocytes. And these merosomes stay intact for a while, and it has a double function of transporting the parasite and actually protecting them from phagocytosis by Kupffer cells. Okay, so back to the skin. Along with the 30% that are leaving with the blood, there are 15% that actually leave by the lymphatics, which we recognize by a slow and lateral movement of the parasite. What is the fate of this parasite in the lymphatic system? Well, they don't go far. We injected the parasite in the foot pad, and all the parasites are actually found in the proximal lymph node, the popliteal. They do not reach the distal one, which is the iliac. We don't find them in the thoracic duct. And importantly, the parasite, in order to be found in the draining lymph node, you can see all these little parasites, they need to be motile. So this is not a simple drainage of the parasite. The parasite actually actively moving inside or within the lymphatics and including in the draining lymph node where they might wreak some havoc. What is the fate of the parasite inside the draining lymph node? Well, most die. At 4 hours, 50% of the parasite are found in association with disease. And at about 8 hours post-infection, we don't see any fluorescence anymore, or just about no fluorescence. Although there are a few individuals that are able actually to invade cells and to start to partially develop in the context of the draining lymph node. You can see here the size at 15 hours, which is the same between a parasite developing in the node and in the liver, and then at 24 hours it starts to go bad. This partial development is always associated with protoplaning cells, supposed to be endothelial cells. So we published these data a few years ago in 2006, and the year after, our colleagues show that after immunization, using irradiated sporozoite and injected into the skin, indeed the CD8 T lymphocytes were primed in the skin draining lymph nodes. So they used a model of P. uelii, which is very closely related to the Bergia that we used in our imaging system, and the bulb C mouse, which is the most susceptible to these parasites. And they used transgenic TCR CD8 plus T cells, recognizing an epitope of the CS protein. And they looked at the number of interferon gamma secreting CD8, and saw indeed that they were only found in the draining lymph node at day 2, and then they migrate secondarily to the liver. So they're primed in the draining lymph node, and then they migrate. They don't go to the liver if you block their exit from the draining lymph node. Even more importantly, ablation of the draining lymph node after immunization decreases the number of interferon gamma CS CD8 cells and abrogates protection in this situation. Okay, so we have 30% that go to the blood, 15% in the draining lymph node. That leaves us with about 55% of the parasite that stay in the skin, and they stay there for hours. It's their final destination. Where do they go? Or what do they become, because that's their final destination? Well, very surprisingly, we found that they can differentiate. And you can see, for example, this individual, that's a time lapse over 4 days of the same site. You can see this parasite clearly increasing in size, and you have all sorts of fates of parasite developing for a while, and then some of them aborting, like this one, for example, that disappears between day 1 and day 2. So if you do a lot of these experiments, you realize, using the model of Plasmodium berghei in the ear, that about 10% of all the parasites inoculated in the skin are actually found developing in the skin. And the proportion, well, slowly decreases with time until day 5. So that's basically probably the same number than the ones that are developing in the liver. The only difference being that the size of these developing parasite in the skin is quite substantially slower, 2 to 3 times, compared to those in the liver. They're smaller, and they indeed give rise to fewer progeny, as we'll see. Okay, so where do they develop? Quite briefly, they develop, and they're found just about everywhere in the skin, and we looked at that through immunohistology of cryosections. So initially, the majority are in the dermis, as you can see here, but they quite rapidly disappear after day 3. At day 3, the maximum, the greatest proportion are found in the epidermis, actually both in the K5 positive keratinocyte and the K5 negative here, so the basal layer or the upper layers of the epidermis, quite surprisingly. And probably even more surprisingly, from day 4 onwards, all the parasites are found associated with the hair follicle. You can see here a parasite, that's a hair follicle, sort of invagination of the epidermis in the dermis, limited by the K5 cells. And in order to confirm that and to be sure that this was reproducible, we did these infections in BLIMP1 GFP mice, in which the cells that are located here just at the base of the sebaceous glands are GFP positive, because these cells that express, BLIMP1 is expressed in progenitor cells of sebocytes. And indeed, there's a constant colocalization of developing parasites with these areas, and they seem to actually persist with time. We saw parasite day 10, day 20. Okay, so the major surprise was to realize that the parasite actually completely developing inside skin cells, and we can see merozoites, which is the progeny of these developing parasites, which normally we only see in the liver. You can see here our merozoites completely filling the cytoplasm of a cell in the skin, and we verified that with both species, Plasmodium berghii, which is known to be quite promiscuous. So we verified that also with P. uelii, that is thought to be really a better reproducer of the behavior of Plasmodium falciparum, which is the main human killer. So both species in rodents actually develop into merozoites in the skin of these rodents. And you remember I showed you the merosomes in the liver, which are these cell plasma membrane bound extensions that leave hepatocytes into the bloodstream, and we see the same, or at least highly fluorescent extensions of these infected cells in the skin, suggesting that the same might be going on in the skin. So, in order to be absolutely sure that we were dealing indeed with merozoites, and that what we were witnessing was indeed reproducing the development in the liver, we did the following experiment. We injected the parasite into the skin, and on day 1, at a time when there is no merozoite that has been released from the liver, we recuperated the infected cell after FACS sorting, as you can see here, and then let them develop in vitro, in vitro conditions. And following these cells, we could see indeed that they were gradually filling with merozoites. And at the appropriate time, we injected these infected cells in mice and realized indeed that these parasites could be able to induce a red blood cell infection. So these little balls that we see inside skin cells are indeed merozoites, and therefore the exact same development that occurs in the liver can occur in the skin, and that's obviously completely novel. Now, we also asked the following question: can skin merozoites generate a blood infection in situ? Because this is obviously not the experiment that I just showed you, and the answer is quite difficult to reach. So we did the experiment using primaquine, which is a drug that kills parasites in the liver. It's a well-known drug. And we used 3 groups of mice. The 3 were injected with parasites in the ear, in the skin. 2 groups were treated with primaquine, supposed to kill the parasites, at least in the liver. Okay, the group 2 and the group 3. And the group 3, we just removed the infected ear. So we eventually have 3 groups of mice. The positive control, which is supposed to have parasite developing both in the ear and in the liver, which is the case. We used a transgenic parasite line that produces luciferase, sorry. Okay, so this is the positive control. The group 3 is the negative control, which is treated by primaquine, and indeed that totally dries up the parasite in the liver. And the infected ear was cut out. And the group 2 is our test group, treated with primaquine and with the parasite in the skin in order to test their capacity to in situ generate a blood infection. The difficulty is that at the dose of 25 milligram per kilogram, this does not completely dry up our negative control group. So there's 1 out of 34 which is infective. And in the test group, we have 6 out of 33. So the conclusion is not straightforward. This is certainly indicative that infection might happen in vivo, but also that it probably contributes extremely little to infection of red blood cell as compared to what is coming out of the liver. Okay, so I'll conclude with this slide. So basically two conclusions. In terms of the life cycle, the picture of the peritransitic phase that we have is obviously more complex than anticipated before imaging. The parasites that are injected in the skin have 3 fates. Obviously, they go to the liver and they will generate the largest pool of merozoites, which will mainly contribute to infection of red blood cells. An important proportion of the parasites stay in the skin, and some will be able to even complete their development, probably yielding fewer merozoites. And it's unclear whether they actually really contribute to blood infection. And if they do so, it's probably anecdotal compared to the liver. Another possible conclusion, which we're very interested in, is whether the parasites that we see persisting in association with the hair follicles are truly persistent parasites. Obviously, you know that the hair follicles with which we see these persistent parasites are immunoprivileged sites. The third fate is towards the lymph node, and that corresponds to just about 15% of the parasites that are injected. But I think that the most important conclusion is probably in terms of immunity, and it shows us that after the natural transmission, including in the situation of injecting immunizing parasites, the sheer mass of parasite antigens is obviously left in the skin. Basically, 15% of the parasite motile, and about 50 to 60% of the parasite that stay in the skin that will eventually die. Most of them will die during the development, and these will secondarily reach the lymph node. That also means that the draining lymph node will be presented with not only sporozoite parasite, but also the antigens, but also antigens corresponding to parasites that are developing intracellularly. So the lymph nodes in the skin will actually be presented with the full array of pre-erythrocytic antigens. So that does not bring us closer to any vaccine, but I think it really gives us a model in which we can really continue to dissect the basis of protection, particularly, hopefully, through immunoimaging. In particular, what are the cells that bring the parasite antigens from the skin back to the draining lymph node, and what are the cells that will prime the CD8. Okay, I'll finish with these acknowledgements, and in particular these 4 people in my lab that have been doing most of the imaging work, the Sepia insect facility platform and the imaging platform, and Geneviève Milon and Christian, so our collaborators. Thank you.